Yes, I'm here to talk about the application of environmental DNA as a potential biosecurity surveillance tool. As Joe mentioned, I'm at the University of Canberra in the Institute for Applied Ecology, and we've just today really just launched our research and development group, which is EcoDNA, which is primarily been funded through the Centre for Invasive Species Solutions, which was the precursor to that was the Invasive Animal CRC. So we've had a bit of a legacy through that and through that sort of partnership, that research and development partnership approach. So what is eDNA, environmental DNA? So basically it is DNA that's left behind in the environment. So sources could be from water, from soil, from air, fecal samples, you name it. We're all leaving DNA behind in the environment. You're all leaving your DNA right now on your chairs and on your glasses, and if we did a forensic sweep, we could probably identify all of you being here. And that's really where the technology has stemmed from, has really been the advances that have come out of the forensic community and in human forensics that enables us now to apply that kind of dealing with really low quantity and poor quality DNA to be able to detect species and individuals within the environment. So it has been super exciting. And that's sort of reflected in that sort of explosion of work and studies that have now shown how effective eDNA is across a number of those sorts of substrates has been in the numbers of publications that have arisen. So really it was kind of trickling along a little bit in terms of, we've always been doing work on bacteria, isolating bacteria from soils and doing DNA from bacteria that comes from out of soils. But it wasn't until it was a publication in 2008, which was the first time where from a water sample, DNA was extracted and a species detected from that. And then another breakthrough happened in about 2012 when Pierre Taboulet, who's a bit of a hero of mine from Grenoble, he developed this DNA meta-barcoding approach, which is, as opposed to doing single species detection from a sample, then you could do multiple species, and meta-barcoding basically means barcoding from a DNA barcode region, but from many, many individuals. So he brought that on board, and as you can see now, and this needs to be updated, but there's lovely, large number of publications and kind of across from water, particle surfaces, and a bit more in air. So the methodology involves from the water perspective, and this is where we kind of started with the invasive animal CRC. So back in about 2013, when I kind of arrived, you probably tell them across the ditch, but when I arrived in Australia, I was sort of thrown into the deep end with this project, and they just said, we want you to have a crack at this. And so given my work previously in kind of forensics and DNA from environmental type applications, this was a bit of a no-brainer. So we had a go, and this is really from water samples. And because the invasive animal CRC at that point were really interested in invasive aquatic species and being able to detect the presence of species that were already existed in Australia or the potential to detect recent incursions. So you kind of take your water sample, you run it through a filtering approach where you filter it onto these cellulose nitrate filters. Then you take it back to the lab and you smush up your filters. As you can see, there's the filters there. They get smushed, extract the DNA from those and through into either quantitative PCR or sequencing, and then bang, there's your answer, which all sounds really simple, doesn't it? So it's making it sound simple, but I'm going to show you a slide which just shows that there was a lot of non-simple aspects to this. So that was all very well and good, and then of course it got me a bit concerned that we needed really to have a solid quantitative framework to be able to utilise environmental DNA. And it got me thinking, if we're dealing with, say, a fish in some water, it's going to be shedding DNA, but there's going to be different rates of DNA production at different times of the year because of what it's doing in the water. Then there's going to be different rates of DNA degradation in that water as well. So sometimes UV breaks down DNA, so even though the species might have been there, it might have actually, the DNA is degraded really fast that you haven't been able to detect it. Then there's things like how many samples do you need to take? So there's the field variables aspect for this. Then there's your laboratory analysis, like the PCR amplifications. How many replicates of all of that do we need to do? So there was a lot of variables in this eDNA detection, and really we needed to create what is a really good quantitative framework to be able to have a detection probability framework. So the sensitivity obviously depends on the amount of water, the number of samples, the number of aliquots, the amount of templated DNA, there's all these things. And then it even gets more nasty and more complicated, and that's when I had to become friends with mathematicians. And because we've got things like dispersion parameters and concentrations in the water, total of molecules across all of our water samples, and then that comes up into this beautiful model, and modelers amongst you would love this, that there's, you can then express it into this equation, which is the probability that no molecules be amplified in a single aliquot versus the probability that the target DNA is actually there. So this was really super important, and this was the foundation of one of the first publications from our group on this work, because it was absolutely essential, because as this kind of stuff goes on. So let's say here we've got a random distribution of our DNA molecules in the water, and so you've got an equal chance of picking up those molecules and an equal chance of extracting it. But for example, and then you're always going to have some variability with your PCR techniques. But then, yeah, there can be clumpiness, and clumpiness, as we've found out, can really be another variable that is across from different types of habitat and can really influence the amount of sort of aggregations of eDNA, because the eDNA itself is actually associated with cells, bits of skin that's coming off, and so that can aggregate in the environment. So we need to kind of get a handle on the clumpiness factor as well. And then what this can do is then develop some models. So if we've got a good modelling framework for estimating that sensitivity, for example here, once we've worked out things like the clumpiness factor, we've brought that into consideration, is this kind of framework where we can work out, right, if a concentration of 10 molecules per litre of DNA molecules, that is the 10 would be the number of samples, and 5 PCR replicates would give you that 95% detection probability. So we've worked out a way of developing our sampling framework for detection probability. So that's been really useful. And then we showed with our kind of target species that we were first working on, which is the European carp, oriental weatherloach and redfin perch. All three of those are highly invasive species in Australia. And you can see the difference there between autumn and spring. Because spring they're a lot more active, they're undergoing, it's probably when reproduction's occurring. The number of samples required to achieve that 95% confidence in detection, for example, oriental weatherloach, you really only need to take about 3 water samples compared to in spring, compared to 16 samples that you would have to take in autumn to achieve that same detection probability. But without knowing that, you wouldn't know how many samples to take. So that's been really the basis to us being able to then deploy this in a really solid framework for a detection surveillance tool. Then we took this to the extreme in Tasmania, where there is an eradication program underway for European carp, and we kind of we said, oh yeah, we'll have a go at this. And of course, I had no idea; I'd never been down there. Went down there, and I was like, oh gee, that's actually quite a big lake, because and I thought, oh goodness, this is a challenge for us. So, for example, Lake Sorell is therefore, is quite a large lake. It's 120,000 millilitres of water, and compared to then Lake Crescent, where it has actually been eradicated. They were in Lake Crescent, but they've been eradicated, which is joined by a small weir. Now, the estimated carp population is somewhere between 1140 and 1840 in Lake Sorell. So we went out there just and had a go with taking 100 samples across the lake, and they've been doing a huge amount of work down there for several years, with large teams of people being deployed, zipping around in boats, using Judas carp, using netting, you name it, all sorts of things to try and eradicate the carp from there. So we got, out of our 100 samples, there were two of those samples had positive for carp, and out of the number of PCR replicates, it was 6 out of 600 replicates, we got 2 positives. And so what that then meant to us is that we could then work out this kind of scenario, which is if, so this is based on the lower population estimate of 1140 carp is the blue line, the upper population estimate is 840 carp. So if we were to say that there were 200 carp remaining in Lake Sorell, how many samples are required to get a 95% detection probability, and that we worked it out to being 7,144. And that's quite a lot. However, cost-wise, that's actually not that bad. So for the amount of money, time and effort, it's about 1/10 of the cost to do an eDNA surveillance program than it is to send a whole teams of people down there and do the work that they have been doing. So it was quite useful from that perspective. So we've done a lot of other subsequent studies and applications. We've looked at things as to how soon can we detect environmental DNA after the introduction of a target species. So we've put poor fish in cages into environments where they've never been and seen how soon we could detect it and how far downstream. We've developed methods for detecting spawning events because different types of DNA get shed when there's spawning versus non-spawning time. So you're going to get much more nuclear DNA versus mitochondrial DNA. So we can look at the ratios of those DNA that can tell us whether something's breeding or not, which is again really important if you're looking at the establishment of an invasive species. We've been looking at the parameters involved for determining biomass for target species, and that's been, we've worked in the National Carp Control Program on that, and we've been trying to use eDNA for the biomass aspects. We've undertaken validations of molecular diagnostics for marine pests, a high priority for Australia, and we've shown again how this method can be really highly sensitive. We've now expanded our assays for a range of non-fish species. We've got amphibians, reptiles, mollusks, crustaceans, all the things that we're working on at the moment. And we've been using high throughput parallel sequencing, so next generation sequencing for species inventories, also from environmental samples as well, as a way of doing biodiversity surveillance in addition to the biosecurity side of things. And now we're also working with CSIRO on investigating using eDNA as a tool for detecting a biocontrol agent within a stored grain pest. So there's a particular stored grain pest which is a snail that has a parasitoid, and the only that was introduced, but the only way to actually detect that parasitoid is that you have to sort of dissect into snails. But what we're doing is using the crushings of the snails that are left behind post-harvest and washing that through water and using that as a way of extracting DNA to detect a parasitoid. So lots of really cool things we can do. So that was a huge amount of fundamental research. What about then turning it into an operational context where it doesn't require necessarily a specialist, lab nerd to do? So in operationalising the technology, we've been working with a couple of companies in Australia, in the US, so Smith, Root and Biomeme, who have been developing with us methods for doing direct filtration in situ, so that you have these the backpack sampling devices for sampling for your eDNA. In the field, you can do the extractions, and you can use your smartphone that you can plug into this Biomeme device, which is basically a quantitative, fancy sort of little handheld quantitative PCR instrument, and you can get a real-time DNA detection in the field. So there are a number of assays now for a particular target species that we can use in that context. Taking it into a larger sort of future-focused context as well. If we look at the future of eDNA sampling and processing, there are a number of different things on the go. So CSIRO have been working on having an autonomous sample filtration archival device that you can just leave out into the aquatic environment, and you can program it to filter a particular amount of water and with programmed intervals of doing that surveillance. So again, quite similar to what Nancy's doing with that sort of passive surveillance with the fruit flies. And then that can feed back, because then also the another device that they're working on can also do the PCR and feed that information back to you. And you could therefore put those things out in arrays in the field, again, very similar to what Nancy's doing. The other device, which up on the top left, is the new device that we're working with, which is for doing multi-species detection and real-time sequencing. And that tiny little device is actually a DNA sequencer. So that's the Oxford Nanopore MinION, and we're one of the sort of first trial laboratories around the world that are using this instrument. And you just basically plug it in to your PC, and with the magic of molecular biology, you can, using microfluidics, put in your sample and get that real-time sequencing. So in terms of eDNA for border biosecurity, we're having a look at different sort of commodity pathways and seeing whether there are certain ones that we could potentially utilise this methodology for. And one that really lends itself to eDNA is the ornamental fish trade. Now, the ornamental fish trade is about $350 million. There's loads of these fish coming in. They're encapsulated within their little bag of water. And so this is ideal in the sense that we could utilise those water samples not only to determine what species of fish are in those bags, but also what pests and pathogens might be coming with them. Because if we're looking at pest and pathogen diagnosis for these aquarium species, even like you really do need to be a very trained taxonomist to tell the difference between something that's totally okay, which is this black tetra, versus a red belly piranha, which is not something we want here. Also, we've found, so Alejandro, who's just joined our lab from James Cook, he had found in his PhD a bunch of parasites infecting these ornamental fish imported into Australia that hadn't been detected as well, because often they don't present themselves as being infectious at that stage. And also, just only last year, an invasive tapeworm was discovered in Perth waterway, which could only have come from a fish, like an ornamental fish that had been brought in from overseas. So where are we at with this, and what are the kind of research and development needs before this actually can be deployed? So we really do need to just continue improving our real-time detection technology to make it usable so that it could be used in those sort of border operations, could be used for different commodities. We're looking at things like, crop washers for the, the brown marmorated stink bug, because that's been another thing that's been shown in the US, that you can actually use that crop wash water to do that. So there's a bunch of things that we're doing to improve that detection technology. The other things that we need are really good reference databases, because although there are global databases that do exist for DNA, unless we have really good targeted reference databases, it's very hard to assign your DNA sequence then back to a species. So that's another big push, and working with CSIRO and collections can help us achieve that. And then from that, and going back to the original sort of quantitative framework that we started with, there is a real potential to be able to establish sort of the standards and guidelines and QA/QC that's required to get the ISO 17025 accreditation, and that's sort of the next step as well, is that we'll be definitely putting together those standards and guidelines in order for this to be operationalised. So if you want to know more, you can contact us at EcoDNA. You can also find us now with our new website as well. Thank you very much.